Okay, welcome everyone. PS Pain Relief Week 4. So this week we'll be covering Epithelium, Biotech and Integrative Metabolism. So I'm Christina, I'll be covering the very first part being Epithelia. So what does that entail? With an epithelia, we'll be running quickly over what primary tissues are, the epithelium being one of them, and we'll also be talking about the general function of epithelia, the characteristics, some types and classifications, as well as specialized epithelial structures, and a quick talk about some cell junctions. So just a bit of recall about cells and primary tissues. Tissues form from cells and tissues will form organs. So the definition for a tissue is a coherent grouping of cells bound together by cell junctions and ECM, which stands for extracellular matrix. It's like this clear fluid jelly and it contains a couple of things. But yeah, what are the different types of primary tissues? First one being epithelium, muscle, nervous tissue and connective tissue. And you've got a couple of examples there and a lovely little diagram you guys can look through with some examples of epithelia, muscle, nervous tissue and connective tissue. Moving on, so what is the general function of epithelia and really what is epithelia? Epithelia is the tissue cells which line the external and internal surfaces of the body. So we're talking, your skin, everything facing the outside as well as inside. So we're talking GI tract, reproductive tract, urinary tract, respiratory tract, and even like the endothelium of our cardiovascular system. So we're talking like veins, arteries, vessels. And what is their function? It really depends on their shape, their structure, physical things about them. So, the number of layers of cells epithelia have, as well as the specialization of the cells. So if do they have cilia? Do they have microvilli? For example, some of them have a protective role, like our skin, epidermis. It's flattened, so it has a high surface area, and it's got many layers. So that means that if your top layers are lost, that's okay. It doesn't really affect the tissue underneath. Other things like absorption, so small intestine in our kidney, these are cells which have microvilli, little kind of hair-like attachments which protrude at the outside of the cell, and that's there to increase their surface area to volume. So it increases their surface area and that helps with their absorptive role. So you can kind of see how structure and function go hand in hand. And last of all, bit of secretion, so small intestine and glands can also be a part of epithelia. Overall though, epithelia have closely aggregated cells. They have really strong junctions, those connections between their cells, all have a little bit of basement membrane or extracellular matrix, which can either be basal or reticular lamina. Those are just some terms you'll hear. And it's a thin layer of fibrous ECM, and it's what separates the epithelium from underlying connective tissue. As you can see in this diagram right here, you've got the basal and reticular lamina, epithelium, and you've got the connective tissue underneath that separates the two. And epithelia are also avascular, which means without vascular vessels, they don't have blood vessels there to supply them, which means for most of their nutrients, oxygen and carbon dioxide, they're relying on diffusion. In terms of the classifications of epithelia, there's two kind of main things we look at. We look at the cell shape and we're looking at the cell type. So shape, it could be squamous, flattened, cuboidal, cube-like, columnar, shaped like a column. And we can also talk about the layers. So is it simple, one layer? Is it stratified, many layers? Pseudostratified, it kind of appears like it's layered based on the positioning of the nucleus, but in fact, it's really just one layer. We've also got more kind of layering types, but it's just extra little categories. First of all, being keratinized, so does it contain keratin? Is it strengthened by keratin? Or it can be transitional. So say like the example being your bladder cells. Those are transitional in terms of the way that they're found because it plays into the role of the bladder and its function where it has to expand in all directions, and that's why the cells are arranged in a transitional different kind of format. Just over the next slide though, we'll be showing like a bunch of images, showing what the different cell types look like and what it might look like on histology. I think you guys will have or would have had an active learning on this already, and hopefully they'll have given you the slides and the questions and the annotations. But here are those images, just going from the left-hand side, you've got simple squamous epithelium. So again, one layer and it's squamous, flattened epithelia. You've got stratified squamous, So squamous, flattened, but in many layers. Simple cuboidal, so cubes in one layer. Simple columnar in one layer. You've got stratified squamous again, yep. And a couple of images. So here we've got some transitional epithelium, and you can see how the stacking and the arrangement's really quite different here. And that's, you know, thinking of the bladder, it's the ability and the capacity for the bladder to expand in different directions, not just unilaterally. But yeah, have a look through some of these images. They are annotated, so that might help you out with just the visualization of what these terms mean and what the cells might look like, and how the most important thing is linking that structure to the function, how those two are really interconnected. Moving on, we'll talk about the specialized epithelial structures and junctions. So this really just, they're classified based on where they're found. Is the junction, the connection between the cells at the top of the cell, on the lateral surface, or basal at the base, and that will determine what they're called. So this is the last little bit about epithelia. In terms of some specialized structures, on the apical, on the top surface, you've got microvilli and cilia. So you'll hear your teachers talking about brush borders being the microvilli. Just as you can see along here, these are the brush borders. And that's there, again, for that absorbative role. You know, you've got actin filaments there, and it's there just to increase the surface area. Whereas cilia, they're a little bit different. They're longer than microvilli, and some examples of where we can find them is that respiratory tract. So you might hear about that mucociliary tract, mucous ciliary, so the cilia. These cells have a lot of cilia, and they're here to kind of form like an escalator and move mucus from our lungs and pathogens we trap to be able to move it up, and then we can cough it out. So that's an example of that structure, the specialized structure being the cilia on the apical top surface, and the function, how it's able to help us, you know, trap and move mucus up that mucociliary elevator. On the lateral surface, talking about a couple junctions. So you got a tight junction, and this is the very highest up, just right here. And zonular adherence, so an anchoring junction, you've got the desmosome and gap junction, as well as on the basal surface, the hemidesmosome. And I think just the main thing which is helpful for me when I was studying this was just knowing what they look like, and I think that was the key part. But yeah, that kind of wraps up epithelium. The next thing we've got is biotechnology.
Okay, so biotechnology. So what's biotechnology all about? Well, we are here to help you guys with that. We've got a box of shapes, barbecue flavor, obviously the best one. And let's get into our topic for today. So what are we gonna go into? So we're gonna go into recombinant DNA technology and therapeutics. I'm gonna go through the basic procedures for creating and cloning recombinant DNA. We're gonna explain the basic principles of PCR technology, so that's polymerase chain reaction. And we're also gonna explain how DNA technology can be used to produce large quantities of therapeutics. My friend Victoria is gonna drop in. She's gonna explain how therapeutic cloning is carried out. She's gonna explain the concept of therapeutic cloning and the different approaches to therapeutic cloning too. So gene therapy, therefore, is our last topic, and I'm going to explain the basic principles of gene therapy, how CRISPR-Cas9 gene editing works, and explain how gene editing can be used for gene therapy. So without further ado, let's get into recombinant DNA technology. Right, okay, so what's the general idea of recombinant gene technology? So the general idea is that I have this one gene that I really, really like, and I want it for myself, right? So how do you get from point A to point B though? There are a bunch of considerations that we have to take into account, and these considerations are what's gonna drive the step-by-step methodology that we're going to follow and that I'm going to take you guys through later. So it's important that we understand why exactly we're doing these things, because if not, we're just, you know, blindly memorizing. So not exactly the best thing. So, okay, I like this one gene, right? But then I have to ask myself, how do I get this gene out? How do I take this gene out of the cell, out of the chromosome that it's in? And then after that, once I have this gene, right, how do we get enough of the gene just in case? How do we make sure that your only surviving copy of the gene isn't going to be destroyed by your body's immune system, by the cells, nucleases that they have floating around, by experimental error, by these kinds of things. How do we make sure that we have the gene intact? And once we know, once we have a secure, supply of the gene, how do we make sure that this gene is supplied to our recipient in a form that's intact and in a form that's usable by our recipient? And how do we then reap the benefits of this gene? Okay, so these considerations, after we've thought about that, then we can move on to really thinking about how we're going to get this done. So this is how we get our we're going to get our DNA from point A to point B. So how do we get this gene out? Well, we're going to use restriction enzymes to isolate our gene. Then after that, how do we get enough of this gene just in case? We're going to multiply the gene using PCR. How do we make sure, therefore, that it's delivered into our recipient? We're going to use the same restriction enzyme to obtain our vector. We're going to combine the gene with the vector, and we're going to insert our vector into the host cell. How do we reap its benefits? We multiply the host cell, extract the proteins, and then turn it into a form that's pharmaceutically useful to us that we can reap the benefits from. All right. Okay. So now that you've understood how and why we do these things, let's get into the details. So first off, we have the restriction enzymes used to isolate the gene. So when we isolate the gene, we do so by using a restriction enzyme that recognizes a restriction site. As you remember from the enzymes topic, restriction enzymes, yeah. Just like any other enzyme, they only recognize one specific substrate or one specific thing to bind to and to transform. So here we see that because restriction enzymes primarily recognize restriction sites, therefore only one restriction site, only one sequence of DNA will be recognized as a restriction site for only one kind of restriction enzyme, if that makes any sense. So if you see EcoR1, it will only recognize this sequence of DNA, and therefore it will only cut at this site to make a staggered end. Okay, so then after we have made our staggered cut with a complementary overhang, we multiply our gene using PCR. So what happens in PCR? Just remember the numbers 95, 55, and 72. Because these numbers are the, I feel like they're the essential part of this process that you need to know. Okay, so at 95 degrees Celsius, what happens? The hydrogen bonds, I almost said hydrogen bomb. The hydrogen bonds between complementary bases are broken. So this turns your double-stranded DNA into two single-stranded DNA strands. And this is followed by annealing at 55 degrees Celsius. So at 55 degrees Celsius, primers anneal to each of the strands. Remember from our DNA replication topic that primers are required anytime you want to elongate or synthesize a complementary strand of DNA with another single strand. So we need our primers here. They anneal at 55 degrees Celsius. And last but not least, elongating. So elongating happens at 72 degrees Celsius, right? So this is, so the enzyme that's responsible for this is Taq polymerase, and its optimal temperature, as you can imagine, is 72 degrees Celsius. So this word hydrophilic, it's not correct. It's supposed to be thermophilic Taq polymerase, right? Because Taq polymerase comes from a bacteria that grows in the vents of like volcanoes and like geysers and stuff like that. So as you can imagine, it really enjoys the heat. It really enjoys being way above normal body temperature. So that's why we have it extending at 72 degrees Celsius. So once this process is complete, we have an exponential increase in the number of in the copies of our gene of interest, because as you can imagine, from one copy we have 2, from 2 copies we'll have 4, from 4 to 8, from 8 to 16, and so on, and you just get a very steep exponential increase And you can calculate the number of copies of the gene you have at the end of this exponential increase by taking 2 to the power of n, where n is the number of cycles that you have gone through for PCR. Okay, so now once we have our gene of interest sorted, this is when we need to obtain our vector. So your vector is going to function as the vehicle that you use to take your gene and put it into a host cell, and then allow the gene of interest to be multiplied by the host cell. Okay, so the vectors that are usually chosen are plasmids, and these plasmids are small circular pieces of DNA. They're usually found in bacteria, and they contain 3 important things that we need for this process. So they contain a replication site. So that means it just codes for the normal process of cell multiplication, you know, mitosis, meiosis. The purpose of the replication site is really just to make sure that if you have this cell, it's not just going to stay as one cell, it's actually going to multiply. Okay, so then your recognition site for your restriction enzyme to cut and to reattach to the gene of interest. So this recognition site, it should be the same recognition site that was used for your gene of interest. So therefore, it means that you have to use the same restriction enzyme that you use to cut your gene of interest and mix it with your plasmids also. And then last but not least is the reporter gene to verify the presence of a recombinant vector inside a host cell. And the importance of the reporter gene is something that we'll get onto a bit later. And in essence, yeah, so you need to use, once again, the same restriction enzyme that was used to make a cut on the restriction site of the gene to make a cut at the restriction site on the vector. Okay, and then you just combine your gene with the vector and then add some DNA ligase so that when the complementary sticky ends do, when the complementary sticky ends do, stick together, they are held tight, and there's no way that they're going to break apart. But as you can imagine, this isn't very reliable because all you're doing is just mixing your gene of interest with your vector and kind of just hoping for the best. So it's possible that your gene of interest, because everything has the same restriction sites, your gene of interest could easily just have stuck to each other and form one large chain of like 24 genes of interest, or 2 plasmids could stick together and form like a mega plasmid or something like that. Not all of the plasmids are going to stick with the gene of interest and form a nicely packaged recombinant plasmid as you see down here. So that's when we need some safety measures. Okay. So these safety measures are going to come in a bit later. But what happens first is that we need to insert the vector into this cell. So we have to heat shock the bacteria. So as you can see, this is heat, and it's also quite a shock. So what heat shocking the bacteria really does is to open up pores in the bacterial membrane to allow the recombinant vector to really get into the bacteria. As you can imagine, again, this is not very reliable because all you're doing is taking very hot bacteria, mixing some of the gene of interest in, mixing some of the recombinant vectors in, kind of giving it a shake and then hoping for the best. Not all of the bacteria will have taken up your recombinant vector. Some of them may have 2, some of them may have 0. So that's why after these 2 steps, which are, you see, step 4 and steps 5, which are not really very, they're not the most reliable. They work on a hope for the best kind of basis. So that's why you need to select the cells that have taken up the vector through the use of your reporter gene. So the reporter gene, it codes for resistance to a specific antibiotic. So what happens is that if the cell has indeed taken up the plasmid contains the reporter gene. That means that the cell that takes up this plasmid is safe from the antibiotic. So what happens is that you literally just plate the cells into antibiotic agar, just kill off as many of them as you can that were unsuccessfully transformed into recombinant host cells. As you can see, it's a fight for survival with all the cells. But then once you get the cells that survive, you just put them into a safe place, put them into a safe environment, allow them to multiply and express the protein through transcription and translation also. So yeah, so at this point, you've basically gotten a nice supply of proteins that have been coded for by the gene of interest that you have nicely isolated and taken out and multiplied. Okay, so this protein is then treated and packaged into a pharmaceutically useful form. So you probably won't just inject the protein directly into yourself. You probably have it in the form of a medicine or a drug or something like that. And yeah, so some uses of recombinant proteins: human growth hormone used to treat dwarfism and hypopituitary deficiency in children, and this uses mammalian and bacterial cells. The tissue plasminogen activator, it converts plasminogen into plasmin to dissolve blood clots. So you guys are going to learn about this much later in the year, probably at the end of Sem 2, but when you learn about the cardiovascular system, you realize that it's very important that we have plasmin in our body, because while it's very important to be able to clot blood when we have injuries, if you just have your blood randomly clotting for no reason at all, this can lead to things like stroke and myocardial infarctions, which are heart attacks. So if you have a blood clot, let's say like, in deep vein thrombosis, this blood clot can travel all the way to your brain and cause a stroke. It can travel to your heart, starve the heart tissues of oxygen and blood, and cause a myocardial infarction. It can also, travel to your lungs, get lodged in one of your pulmonary vessels and cause a pulmonary embolism. All of these, not fun times, really not fun times. So that's why you need tissue plasminogen activator to make sure that you have a steady supply of plasmin, to make sure that you never have any of these unnecessary blood clots just kind of plugging up your system and causing all sorts of mayhem. So E. coli cells are used to synthesize tissue plasminogen activator. And I mean, last but not least, you have insulin also. You really don't need to memorize a lot of detail on what insulin does. You do need to know what insulin does. You just don't really need to know exactly how it's made. Just know that you also use recombinant gene technology for insulin. Okay. And next up is therapeutic cloning. I'm going to step out for this one. My friend Victoria is going to take over, and I'll join you back in the next topic. So thanks.
All right. I'm going to keep talking about therapeutic cloning now. So therapeutic cloning uses modified cells to grow healthy patient-specific tissue. And it grows embryonic cell lines with the same genome as the nuclear donor. So this will make more sense in the next slide coming up. So, so it creates patient-specific cell lines for differentiation into specific tissue types. And overall, it reduces the immune rejection because cells are from the patient, not from like, say, for example, a mouse. So therapeutic cloning can be achieved in two ways. The first way, or the main way that's probably most relevant to you, is somatic cell nuclear transfer, which is short we call SCNT. And that, again, creates patient-specific cell lines that differentiate into specialized tissue. Or the second way is reprogramming of adult cells to form induced pluripotent stem cells, or iPSC, which can then form specialized tissue. So therapeutic cloning is all about creating patient-specific tissue, okay? So therapeutic cloning works on the basis of cell potency. So you've got three levels of cell potency. So you have highly potent cells, which are totipotent. and they can give rise to all embryonic and extra-embryonic cells and tissues. So your zygotes, which is essentially the first point of fertilization. When an egg is first fertilized by a sperm cell, you get a zygote, as you know, and this zygote is what we call totipotent. It is the highest level of potency, and it can give rise to all embryonic and extra-embryonic cells and tissues. So if you look over here in this little diagram, this is where the zygote would be, and see how it says totipotent here. So these can give rise to all other cells, okay? The next level of potency is pluripotent cells, and this is what you're going to be mostly concerned with when it comes to therapeutic cloning. So your pluripotent cells can form all somatic cells. So for example, the embryonic stem cells from blastocysts are pluripotent, so they can give rise to all tissues, basically. They just can't give rise to extra embryonic cells and tissues. The reason why we're concerned about pluripotent cells is because if you think, for example, we're trying to replace damaged tissue in the heart after a heart attack, okay? You want to be able to create cardiomyocytes from this level. So they're at this level. They're not embryonic cells, right? So we don't need to go back to the totipotent stage if we just need the pluripotent level. And then finally, there's multipotent, which is limited potency, and they can only give rise to cell types of their lineage. So less important, just remember our pluripotent cells, which is your blastocyst, which contains embryonic stem cells that can give rise to all these cells down here. Okay, so the first type of therapeutic cloning, somatic cell nuclear transfer, or SCNT. Let's just go through the process first of all. So first you remove the nucleus from a recipient egg, okay? This will give you a kind of like a blank slate. It gives you an egg without a nucleus. And then you extract a nucleus from a somatic cell of the patient, okay? So this nucleus is now important. You need to remember that the egg doesn't come from the patient, okay? The egg comes from any other donor. The nucleus is what is important and needs to be patient specific, because you're trying to make specific tissue or cell types for whatever reason for this patient. You need a nucleus from their cell, and that's how you're going to create patient specific cell lines for them. Yep, okay, so the patient's nucleus is inserted into the recipient egg. So now the egg contains the patient's genetic material, okay? The egg then divides and it forms a pluripotent blastocyst, which is an early embryo. And then from there, you can collect embryonic stem cells from that blastocyst. So see here in this diagram on the right, this is your blastocyst. You've got the inner cell mass, which has all the embryonic stem cells that you can now collect. And then the embryonic stem cells can then be cultivated and induced to differentiate into specialized tissue for that patient. So again, in this diagram, you've collected the embryonic stem cells from this blastocyst that you've created. You basically induce it to form different cell types for the patient. So you can form bone tissue, muscle tissue, or nerve tissue, and that can go into that patient as needed. So why somatic cell nuclear transfer? This form of therapeutic cloning has the potential to treat diseases in any organ/tissue by replacing damaged and dysfunctional cells. So after a heart attack, for example, okay, you can have cells dying in the heart, which you can imagine to be quite detrimental. And what SCNT can do is replace those damaged cells, because you're essentially recreating that cell again, okay? And then you can also, as I said before, diminish the risk of immunological rejection because the cells are patient specific, and you can dramatically reduce the wait times for organ transplants, okay? Because you can imagine that you don't need to wait for donors or donor organs to be available. You can just create it in the lab. The challenges of SCNT, however, is that it requires the destruction of human embryos, okay? So after you're done collecting your patient-specific tissue, you have to destroy the human embryos, and that is obviously an ethical problem with SCNT. So then your alternatives to using embryonic stem cells, you can use adult stem cells, which is what we're going to be talking about next. And then you can convert these, or you can convert somatic cells to cells that have stem cell properties, all right? So that leads us into the next way that we can carry out therapeutic cloning is induced pluripotent stem cells, okay? So induced pluripotent stem cell therapy, you can take normal adult cells and you can modify them and induce them to form pluripotent stem cells. These iPSCs can then be induced to form specialized tissue. So here, again, it's just another diagram showing the cell potency. If you look here, this top part shows your SCNT method. If you go down the line, we've got cells that are already part of their specific lineage, but you can actually go backwards and induce them to form pluripotent stem cells again, and to which they go back to the pluripotent stage. So one way we can get induced pluripotent stem cells, right? This scientist, he took stem cells from the nose, okay, because in an adult body, we still have stem cells that can give rise to different cell types. So this scientist, he took stem cells from the nose of people with paraplegia after a traumatic spinal cord injury. He then grew these nasal stem cells in the lab. Once they were grown, he transplanted these cells back into the patient's injured spinal cord. Okay, so it's the same patient each time to aid in repair regeneration, and it worked. So this trial provided a precedent for future trials using adult stem cells. Okay, so here we're looking at adult stem cells in particular. The other way we can get iPSCs is by reprogramming normal adult cells. So we can reprogram adult cells that are not stem cells, okay? So basically in an experiment, they found 4 genes that coded for specific proteins, and these proteins control genetic switches, and then these were then transferred into like normal skin cells that weren't stem cells. And then these proteins triggered expression of other genes that then led the cell to become induced pluripotent stem cells. Okay, so that's just this diagram here. You've got non-stem cells, so normal skin cells are removed from the patient, a vector carrying the genes are added, and then they grow into a mass of pluripotent cells. As always, there's always challenges. So the challenges of iPSC, treatment using these kinds of cells are not always successful. You can get epigenetic differences. So induced pluripotent stem cells are not reset to embryo-like, near tips and centers of chromosomes. There's additional methylation. So it's not the exact same as the embryo form. And we're not sure what this could mean in terms of how it would actually play out in a patient. There's also genomic abnormalities. So iPSCs can incorporate extra cancer-causing genes and fewer tumor-suppressing genes. So you can imagine that would be another concern of using iPSCs. So finally, our understanding of the pluripotent state is still not strong. So all of this is very new kind of territory for us. And yeah, that's it for therapeutic cloning.
Okay, so hope you guys enjoyed that bit on therapeutic cloning. Thank you, Victoria, for picking that one up. So now I'm back to tell you guys about gene therapy. Okay, so gene therapy basically is inserting a normal allele into a specific diseased cell, allowing the diseased cell to begin expressing the normal proteins instead. So instead of you know, in the previous sorry, not the previous one, but in recombinant DNA technology, you insert the proteins into you. instead of inserting the proteins into your cells, you insert the gene into your cells and you allow the cells to begin expressing normal proteins on their own. Okay, so basic steps of gene therapy. Honestly, this diagram here is all you need to know. So you see, we have the gene here. It's isolated. This gene is inserted into a viral vector. So this virus carries the normal gene, right? It carries the normal gene into a somatic cell. And this somatic cell, therefore, is kind of infected by the virus. You guys will learn later on that viruses can insert their own genomes into human genomes, and that's why they're so damaging, because they can kind of like take control of our cells and make them either stop defending themselves or stop defending themselves, or stop killing viruses, or just stop functioning altogether. But here, this capability of viruses is used in a positive way. So viruses can insert its genome that includes the normal gene or the healthy gene into our human genome, into our human cell genomes, and patients can express the normal gene and produce normal proteins. All right, so okay, but this is merely an addition and not a correction or replacement. So if you have a faulty gene, all this does is just kind of like stick in a normal version of that gene, but it doesn't take the unhealthy gene out, right? So this, as you can imagine, is not a very efficient process. It's not a very effective process. And also, because viruses, they don't have a sense that tells them like, oh, I'm going to insert this gene in a place that matters, or I'm going to insert this gene in a place that is useful. So these viral vectors are just going to stick the genes that they have just anywhere that they want, right? So that means that the insertion of this normal gene is random, and it can mean that if you have certain cancer-preventing genes, again, you'll learn about that I think in a few weeks' time or something like that. But if you have a gene that consistently that codes for proteins that hold off the formation of cancer, and you stick a gene right in the middle of it, right? Then this means that your cancer-preventing gene is going to be disrupted, and you're going to have your defenses against cancer are just going to be lowered, and that's going to spell trouble for you. So therefore, this is limited in its efficacy for gene therapy. So this is where gene editing comes in. Yeah. All right. Okay. So gene editing. What is it? It's altering the genome of a cell using the natural self-defense mechanisms in bacterial cells to repair defective DNA. Right. So this is done using the CRISPR-Cas system. So the CRISPR-Cas system is really, we have to give credit to the bacteria for this system, because it's present naturally in them, and it sort of functions like the human adaptive immunity system, which you guys, again, are going to learn further on in your journey. But basically what happens is that the CRISPR sequence is a sequence in the bacterial genome. It contains fragments of viral DNA that have been incorporated into the bacterial genome. So when a virus invades a bacteria, this bacteria can kind of like steal or kind of just grab fragments of the viral DNA and incorporate it into its own genome. So this actually helps to give the bacterial immune system a memory of the viruses that have previously invaded it. So in the event of a viral infection, what happens? So this sequence of DNA, the CRISPR sequence that contains a piece of viral genome, it's transcribed into RNA, right? So this becomes guide RNA. So guide RNA, what happens to it is that it's used to compare. The bacteria uses it to compare it to viral attackers. Sort of the way, you have like wanted posters, right? So if you see like oh, look, it's a predator. And the photo on this wanted poster looks exactly the same as this creepy old guy. He's probably a predator. Let's arrest him, right? So in the same way, the Cas9 enzyme, it pairs up with the it pairs up with the guide RNA. So if a sequence of viral genome happens to match to the guide RNA, as you can see in this picture here, then the Cas9 will know immediately that they have an interloper on their hands, and the Cas9 will proceed to just snip up the viral genome and kind of prevent the viral genome from functioning anymore. So essentially killing the virus. So as you can see here, once again, this was supposed to be a joke, but I'm realizing it's actually pretty helpful. I don't know if you guys feel like that, but I'm thinking that it's actually pretty helpful. So the second a guide RNA matches a viral genome, an external viral genome, Cas9 just comes in and it breaks down the door and it just cuts up the viral genome and kills your virus. So yeah, so that's how it functions in nature. So how does it function as a therapeutic procedure? So what happens here, let me just move my person bubble. Okay, so what happens here is that the CRISPR sequence now is made to contain a nucleotide sequence that matches the patient's defective gene. So if you have, let's say, an insulin-producing gene that is not working, the CRISPR sequence will now have a sample of your faulty gene, right? So this segment is then transcribed into RNA. So now it becomes guide RNA. So now that you have guide RNA that matches your faulty gene, Cas9 combines with the guide RNA. You can now tell where your faulty genes are, and it can go in and cut your faulty gene out. So this means that now your faulty gene has been removed from your genome. But like, what happens now? So do you just not have a gene? Well, okay. So repair sequences are also introduced. So these are basically copies of the normal gene. So once the defective gene has been cut out, we introduce repair sequences into the cell. The cell has natural DNA repair mechanisms that will kind of repair the cut that was made, and they'll just insert the normal gene, right? So then, as you can imagine, that's the end of that. It's very, very precise. It's very targeted, and you make sure that you fully take out the defective gene. And this can be used in beta thalassemia and retinal diseases. Again, details of which are not super important. So yeah, that's the end of that. Thank you so much for tuning in. Next up, we have integrative tissue metabolism. So good stuff.
Alright guys, good news, we're finally at the end of metabolism. So today we're going to go over integrative tissue metabolism. We've learned how carb, lipid, protein metabolisms occur, and now we're just wrapping it together and linking it to all the different tissues. So the four major organs of fuel metabolism are the liver, adipose tissue, muscles, and the brain, and they communicate with each other using hormones, the nervous system, and circulating metabolites. And the substrates used by one pathway may be the metabolite from another pathway, but this just means that all these different pathways are interconnected. So yeah, there are three states of the body. There's the fed state, the fasting state, and starvation. So the fed state obviously is right after you eat. It's when your body is digesting food and absorbing the nutrients. And when the pancreas detects an increased level of glucose in the body, it releases insulin. So this is the signaling molecule that signals all of these different organs to do different things in the fed state. And the fasting state is between meals, when the food is mostly digested, absorbed, and already stored. So this is like overnight when you're sleeping. And this is associated with glucagon release by the pancreas.
And once again, it's a signaling molecule to tell all these different organs what to do. And it's kind of like the opposite of insulin. And lastly is starvation. So this is beyond the fasting state. It's when your body is pretty much running out of, you have eaten for a long time and your body's running out of all these energy sources for a long period of time. Okay, so obviously the liver is at the center of this all. In the fed state, Norma likes to call it the lipogenic liver because it's focusing on forming fat for storage in the body. And so in terms of glucose, let me get my pointer out. In terms of glucose, obviously any glucose in the body is used by all the different tissues for energy synthesis. And hepatocytes, liver cells, convert any excess glucose to G6P. So this commits to it becoming used for the synthesis of glycogen. And this is the process of doing this also maintains the concentration gradient of glucose in the blood, so that the glucose, instead of keep going into the liver, it knows to move on to other cells where it's needed. So yeah, there's also increased glycogen synthesis for energy storage. In terms of amino acid, from the protein that you eat, it is converted to ketone bodies that can later be converted to acetyl-CoA and used for a variety of other things, because acetyl-CoA is pretty flexible. And in the fasting state, the liver is known as the glucagonic liver, and it focuses on maintaining the blood glucose level. So in terms of glucose, we stop synthesizing glycogen, and instead we're breaking down the glycogen to release glucose to maintain the blood glucose concentrations. And after this goes on for a while, we have gluconeogenesis. So now we're making glucose. So we do so to replace the blood glucose and also the depleted glycogen stores in the liver. And in terms of lipid, ketone oxidation can release lipids and glucose for use of ATP. And so once all these excess glucose has already been replenished, they can also be converted to tags, triglycerides, and fatty acids for long-term storage. And lastly, in starvation mode, we'll keep coming back to this. Basically, a lot of metabolites from the muscle can be sent to the liver for gluconeogenesis to maintain glucose. And the liver can also generate ketone bodies that are used for the brain and heart. So we'll come back to this later as well. So skeletal muscles, as we've seen in the great metabolic race from last week, the muscles can use pretty much any energy source. You can use free fatty acids, ketones, glucose, creatine, phosphate, and this depends on the degree of muscular activity. So I really recommend you, if you're not familiar with this, go back to last week and look at the great metabolic race. And skeletal muscles accounts for about 20 to 40% of the oxygen consumption in the resting body, and up to 90% when we're in active muscular contraction, like when you're exercising. So in the fed state, glucose is the primary source of fuel. So muscle cells would take all these glucose to either use energy or to synthesize glycogen to store in the body for later use. Lipids are of secondary importance. And also in the fed state, amino acids are taken into muscles to build more muscles, basically. So for protein synthesis to replace degraded muscles. In the fasting state, this amino acid can be used to generate energy in the liver through gluconeogenesis. But you'll see that so like when we're talking about starvation, it really is only when all the energy stores of the body are fully depleted that the proteins in the muscles will be are released and broken down for gluconeogenesis to become the primary source of energy for other tissues. And the reason for this, obviously, is that protein is really important for the body and it's important for our muscles. So our body tries to use everything else before it uses the proteins. And now we're going to, all right, so I'm going to move on to adipose tissue. So adipose tissue are where all the energy in the body is mostly stored. So they're stored as in the form of tags. And there's an adipose site right here. And you'll see that all the organelles are pushed up to one side so that the main cell is occupied by like a big fat droplet. It's located over the body and it's very metabolic active. So in the fed state, all the glucose, excess glucose in the blood is sent from the liver to these adipocytes to promote tag synthesis for storage. And all the fatty acids, straight from our diet, are brought from chylomicrons and then they're broken down into fatty acids, and they enter these cells where they're also used for tag synthesis for storage. In the fasting state, these tags stored are mobilized and transported to tissues such as muscles and heart, and where they're just used for energy production. And the glycerol in the tags can also be used by the liver for gluconeogenesis to maintain blood glucose between meals. But in starvation mode, these fatty acids can be converted to acetyl-CoA, once again for energy synthesis. And then, once again, the glycerol can be used for gluconeogenesis. But most importantly, the fatty acids in tag stores can be used to make ketones. And ketones can be used by some specific organs, such as the brain and the heart, to prevent continued breakdown of proteins that is safe for gluconeogenesis when we really, really need it at the end. And I'm just going to move on to what the heart, the brain, and red blood cells use as energy sources. So in the heart, the heart primarily relies on fatty acids because it can't really store any lipids or glycogen. It needs to be continuously active, unlike skeletal muscle. And as you can see here, it can also use glucose and ketone bodies if it needs to. And it only relies on aerobic respiration. You can probably imagine that when the heart is lacking oxygen, it would cause a heart attack. So that's why I always use aerobic. And in starvation mode, can use ketone bodies. So the brain primarily relies on blood glucose. It consumes 20% of oxygen in the body. And it doesn't have any fuel reserves at all, thus it needs a constant supply of this glucose. And since you can probably guess the brain is important for the function of the entire body, so it normally gets special priority in terms of the supply that it gets. And in starvation mode, it can use ketones like the heart. So ketones rarely cross cell membranes, they can enter the brain. And for both cases, the use of ketones for both cases, as in the heart and the brain, being able to use ketones means it takes burden away from the liver to have to degrade proteins and use it in gluconeogenesis. And lastly is red blood cells. So red blood cells only uses glucose and only responds anaerobically because it doesn't have any organelles to do anything else. It uses both glycolytic and pentose phosphate pathways. It uses a lot of glucose because it's inefficient. It only uses anaerobic respiration, and as you know, glycolysis only results in 2 net ATP at once. So it uses a lot of glucose and has the highest specific rate of glucose utilization of any cell in the body. So lastly, we're going to look at 2 diagrams, and they're kind of just a summary of everything we went through besides cardiac muscle and red blood cells. The left side shows the body during the fed state, and the right side shows the body in the fasting state. Let's start with the fed state. So, like I said, after a meal, when blood glucose is high, the pancreas secretes insulin that signals to all these organs to do what they're supposed to do after you eat. And from the intestine, you can see all these nutrients that you just ate and where they go. So we'll start with glucose. Glucose goes through the blood and goes to your liver. So first of all, these glucose can just be used to send to other organs for energy synthesis. So glucose can be sent to the brain, can be sent to the adipose tissue to be stored there, or it can go to the muscle to be used. Oh my God, stop moving. Stop. Okay. And amino acids and from the protein that you eat can undergo different things. First of all, it could just be excreted as urea. It could be used to make ketone bodies, or it could be used in protein synthesis. And then the fats, it goes through the lymphatic system, and it could just be delivered to the liver and then to fat tissue for storage, or these fatty acids can go to your muscle and be used as an energy source as fatty acids. Okay, now we're gonna move on to the fasting state. So when the pancreas detects that blood glucose is dropping, it releases glucagon. So this hormone signals all the things to all these organs of what they should do in this situation. So where should we start? Well, obviously the most important thing is that glucagon signals for glycogen to be mobilized. So glucose to be mobilized from glycogen, and it becomes G6P, and then this glucose can be used for the brain. Or, for example, in the muscles, the proteins can be sent to the liver, where it's broken down to amino acid, and amino acid can become pyruvate and be used in gluconeogenesis. Or let's look at fat now. In tags, you know how tags are made of fatty acids or glycerol? Fatty acids can be used in the liver just for energy synthesis, like this, or it could be used to make ketone bodies that can be used by the brain in starvation mode. Or these ketone bodies can also be used as in the muscles as an energy source. And as for the glycerol can be used in gluconeogenesis to make glucose again, and G6P can be used by the brain. And I think that's everything. So thank you very much.